for giving me an opportunity to have a presentation here today. I'd like to talk about the climate change and emerging infectious virus, what we do at the NIH. Recently, Gavi and IPCC reported 5 key links between climate change and health. One, greater spread of the insect-borne disease. Second is the water and food scarcity can mean malnutrition and greater disease. Third is that increasing poor air quality can cost lives. The first is that displacement and migration and putting people at risk. And 5, climate change is affecting mental health. All these factors affect the human health and immunity. Okay. As the weather warms up in Korea, the range of vectors can transmit disease expands. Infectious disease previously limited to southeast also get upwards to the northern countries. The number of imported, the vector-borne illness, dengue, Zika, chikungunya increased in Korea. Therefore, at the national level, countermeasures such as therapeutics are needed to prevent the import of the vector-borne infectious disease into the country and to prepare for outbreaks. South Korea government has made it priority to proactively manage the impacts of climate change by developing and implementing adaptation policies. These policies aim to mitigate the effects of climate change, including the responding to health issues, natural disasters, and other impacts. Since 2008, the Minister of Environment has been working with the 17 ministries to plan and implement climate change adaptation measures every 5 years. The Korean government recently published the 3rd National Climate Change Adaptation Plan. There are a total of 341 projects and policies aimed at mitigating the effects of climate change in the field of the water, ecology, land management, agriculture, fisheries, health, and industry, and energy. Some priority risks related to health and especially infectious disease have been identified, including increased vector-borne disease, waterborne illness, and emerging infectious disease. To protect public health from the infectious disease caused by climate, the Korean government have focused on the R&D of acute and chronic infectious disease and the management of the infection event-based surveillance, EBS, water and foodborne illness surveillance, Internet, and vector-borne illness, VectorNet. If you want to see the data, you can visit the website of KDCA. I will now provide the detailed information on the research of development of acute and chronic infectious disease. Sorry. Korean Disease and Prevention Control Agency, KDCA, has conducted the R&D programs to study vector-borne infectious diseases that are relevant to climate change. The program includes the research on the pathological, molecular, and genetic studies of vector-borne infectious disease associated with climate change. KDCA also studies the ecological characteristics, roles, and transmission potential of infectious disease vectors such as ticks and Aedes aegypti mosquitoes. One of the program objectives is to develop therapeutic technology for vector-borne disease and test their efficacy. At the KNIH, Center for Emerging Virus Research, we have developed and tested antivirals and monoclonal antibodies for treating SFTSV and dengue. In May of this year, the Korea KDCA released the mid to long-term planning for the emerging infection pandemic as a national pandemic preparedness and response plan. I have compared this plan with some programs implemented by the NIAID of USA. After the COVID-19 pandemic, NIAID has managed the AVID, Revamp, and NextStem project to prepare for the next pandemic. AVID focused on the evaluation platforms for discovering small molecular antivirals targeting pandemic potential RNA viruses, including the seven different virus families. Meanwhile, REBAP and NextGen have managed programs to develop vaccines and monoclonal antibodies against these viruses. On the other hand, KDCA's mid to long-term planning for the emerging infection pandemic aim to reform the R&D support system for developing vaccines and therapeutics against these 8 viruses. Our first step in this plan is to obtain therapeutic libraries through the use of repurposing broader range antivirals, including nature product-derived compounds and monoclonal antibodies. Additionally, we plan to implement a high-throughput screening system and new technology to accelerate discovery of antivirals. Our ultimate goal is to enhance our response to infectious disease through active collaboration and reach clinical phase 1. Many scientists have emphasized the importance of developing vaccines, antivirals, and diagnostics to prepare and respond to the next pandemic. While the development of vaccines is essential, some pathogens are hard to vaccinate against, and some vaccines demonstrate limited effectiveness, and some vaccines are not suitable for certain populations, and some individuals may refuse to be vaccinated because of their concerns about vaccine safety and believe that infection is not a serious threat. So, therefore, antiviral drugs play a vital role in planning for and responding to future pandemics. My second part of my talk is I will discuss about the development of antivirals at the Center for Emerging Virus Research of NIH. We have discovered several antivirals through the intramural research and global collaboration and extramural research. These antivirals are effective against MERS-CoV, SARS-CoV, SFHSV, influenza, dengue, and Lassa virus. Our MERS-CoV therapeutic monoclonal antibodies is currently in the preclinical phase, and the Division of Acute Infectious Virus Research is screening to discover monoclonal antibodies of RSV, and our global collaborator in Chile is also screening to find nanobodies against Lassa viruses. Additionally, we are planning nanobody research to obtain neutralizing antibodies against the Nipah virus. As I move forward, I focus on the importance of antibiotic therapeutics. Some people may be doubtful about the developing antibody treatment for infectious disease because of the rapid mutation of RNA viruses. However, in the case of COVID-19, it took almost 2 years for medical medicine to be approved. Meanwhile, therapeutic antibodies were rapidly developed and approved during that period. And many patients worldwide were successfully treated. Therefore, it is crucial to develop antibody therapeutics promptly when a new infectious disease emerges. Additionally, some oral antiviral agents can be genotoxic since they are type of nucleoside analogs. On the other hand, human monoclonal antibodies are the most natural forms of antivirals that use the human immune system. Hence, antibody therapeutics are safer and more effective in responding to emerging infectious disease than other modalities. I'd like to talk about rapid antibody technology briefly. Rapid antibody technology has made significant advances since the early 2000s. The antibody page display system is a well-known tool for quickly discovering high-affinity antibodies. By using page display system, we were able to find neutralizing antibodies for SARS-CoV-2, such as CTP59 and for SFTSB, GNGC. We also used the humanized transgenic mouse to screen for neutralizing antibody against RSV. And NIAID introduced a technique that constructs an antibody from a single B cell sorted by structure-based designed viral antigen probes. The technique can save more time than antibody development using a classical method such as mouse hybridoma. We used the method and discovered several neutralizing antibodies, MERS-CoV and SARS-CoV and SFTSV antibodies. We were able to discover 6 potent neutralizing antibodies thanks to the MERS-CoV spike trimer protein supplied by Dr. Barney Graham. And additionally, we found 4 highly potent neutralizing antibodies at KNH with the IC50 activity at the nanomolar level. And 90 F1 antibody demonstrate the highest neutralizing activity in both in vitro and in vivo transgenic mouse models. Our successful collaboration was published in PLOS ONE article when we obtained a joint patent. The X-ray crystallography-based structure character regions of neutralizing antibody 90F1 complexed with RBD revealed the 3 critical epitopes in the RBD region of the spike protein. We tested the therapeutic efficacy in DPP4 transgenic mouse, which expressed the MERS-CoV host receptor human DPP4, even at the lowest dose of 0.156 milligram per kilogram. The KNH88 antibody treatment results in 100% survival of the mouse group. To evaluate the prevention efficacy of these antibodies, we injected them via the IP route 1 day prior to the MERS-CoV challenge. All the mouse groups injected with the KNH72, 88, and 90F1 at the lowest dose exhibit completed survival. Through the collaboration with the Celtrion, a highly potent neutralizing antibody to SARS-CoV-2 was developed using the page display system, resulting in the 3rd COVID-19 treatment approved worldwide and the first approved in Korea. In 2020, due to the COVID-19 pandemic, our manpower was focused on assisting and supporting Celtrion, which results in the late development of monoclonal antibodies at KNIT. In 2021, with the emergence of the SARS-CoV-2 variants such as Alpha, Beta, Delta, Gamma, and Omicrons, we began looking for broad-range antibodies capable of blocking them. Fortunately, we found 2 broad-range neutralizing antibodies. We converted both the antibodies from the IgG forms into the IgA forms, which results in stronger neutralizing antibodies. In fact, the neutralizing antibodies of both IgA antibodies were 6 to 1,000 times stronger than their IgG forms. As I mentioned before, the climate affects the vector-borne illness. The Asian longhorned tick is the primary carrier of SFTSV, a virus that causes severe fever with thrombocytopenia syndrome. SF-TSB RNA has been found in various tick species in the area where SF-TS is prevalent. The Asian longhorned tick is native to the Asia-Pacific region, including China, Japan, and Korea. It is known to be fed on both wild and domestic animals. Case of the SF-TSB infection has been reported in Vietnam, and since 2017, the tick has also been identified in the US. However, SF-TSB RNA has not yet been detected in the ticks found in the US. Climate change may cause the ticks to spread to more parts of the world. Treatment of SF-TS infection is important to survival and disease control. Many studies show that several agents have the therapeutic effects in vivo mouse and hamster models with SF-TSB. However, there is no clinical trial to prove ribaverin's beneficial effects in treating hospitalized patients with SF-TS. But studies have shown that favipiravir might be more effective than ribavirin in treating patients with SF-TSB. However, currently no specific antiviral treatment is approved for the SF-TSB, and recovery mainly depends on the supportive treatment. As one of the projects of National Climate Change Adaptation, KNIT identified 3 flavonoids that inhibit SF-TSB replication. Among them, gomycin G was more potent than favipiravir with no cell cytotoxicity. Several monoclonal antibodies have been identified for neutralizing effect against SF-TSB. Among them, MAP45 is discovered human neutralizing antibody that recognized and terminal region of SF-TSB gene glycoprotein. However, it has only been shown to have a neutralizing effect in vitro, and its efficacy in vivo is not yet to be determined. Another monoclonal antibody, F10, has been promising therapeutic effects in a mouse model of SF-TSB infection. F10 binds to domain 2 of GN. Furthermore, camel nanobodies have also been isolated to develop neutralizing antibodies against SF-TSB, SNB02. To better explain the development of neutralizing antibodies to SF-TSB, it is important to take a closer look at the GN and GC antigens of SF-TSB. The middle segment of the virus genetic genomes encodes a precursor polyprotein called GN-GC, which gets cleaved by the host cell proteases. When SF-TS enters the cell, it does so via receptor-mediated endocytosis. GNX acts as an attachment for the virus to host cell receptors, while GC fused with the cell membrane after GN has attached. There are several host receptors for SF-TSB can target, including GC, SINE, L-SINE, NMH2A. We will adapt 2 strategies to our project. First, we screen antibodies using phage display system. And second, we are screening antibodies from B cells of patients who have recovered from SF-TSB. We obtained antibodies with high affinity to GC antigens at the picomolar level through the phage display system. However, we found this antibody did not have any neutralizing antibody. Despite this, we discovered these antibodies could detect various SF-TSB genotypes, so we can decide to use them as a diagnostic antibodies. We recently isolated the B cells that bind to both GN and GC probes and need to examine the characteristics of these antibodies. Since genes attach to many receptors expressed in the B cells, it was very difficult to differentiate the B cells with the gene-specific IgGs from those having these receptors. However, we also discovered GC-specific antibodies that have a mild neutralizing antibody. We conducted studies to combine antibodies and found that their synergic effect enhanced their neutralizing antibody. I want to share the diagnostics we developed at our center. We generate real-time artificial diagnostic for MERS and 7 hemorrhagic fever viruses, which were then commercialized through the collaboration with the other companies. Additionally, we discovered monoclonal antibodies targeting nucleocapsid proteins of MERS-CoV, SARS-CoV-2, Nipah virus, and SFTSV. These antibodies are used in serological diagnostics, ELISA, and rapid kit. To develop a sandwich ELISA test for Nipah virus, we conducted computational assays to identify optimal candidate combinations and conditions. And we also found the MERS-CoV nucleocapsid monoclonal antibodies from the page display systems. This is the last one. Our future plans involved developing new technologies and platforms for rapid antiviral development to better prepare and respond to emerging infectious diseases. Some of the key areas we are focusing on include the nanobodies and RATP-IG, antibody engineering for long-acting antibodies, AI-based drug design, and high-throughput screening system. And collaboration is also essential for rapid response to the next pandemic. We need to proactively manage our reagent portfolio to produce antiviral agents in GLP or GMP level infrastructures to fast-track FDA approval. We plan to collaborate with Korean bio companies that have GLP and GMP infrastructures and obtain chemical libraries, natural libraries, and antibody cell bankings. Finally, we'll focus on the commercializing the therapeutics and diagnostics, as it is crucial to ensure that our efforts translate into effective treatment options for those in need. Our plan includes expanding collaboration networks at domestic and global level, ranging from basic R&D to clinical research. To prevent the control highly pathogenic infectious disease, it is crucial to collect clinical samples relevant to pathogen not imported to Korea from global networks. We aimed to expand in vitro and in vivo screen services for companies collaborating with IPK and Korean University to develop antivirals. This will help protecting public health from emerging infectious disease and climate change. Thank you for your listening.
Any question? Dimitri?
Yes, thank you very much for this talk. As you know, the problem with emerging viruses, like you mentioned MERS or other emerging disease, is capacity to do some clinical trials. So do you have any partner you are thinking to make some clinical trial already for MERS? Did you have interaction with Middle East country, for example, already, or do you plan to test any?
Yeah, it is very difficult to collect the clinical trials. So we need to collaborate with the global networks, especially Middle East Asia. So we try to connect to the Middle East Asia Institute. Yeah.
Another question.
My question is a quick question. just to know if you think your research, if you can apply some aspects like data-driven in your research, it can be helpful and improve your research in that field.
Excuse me, could you please explain more?
Data-driven research.
Data-driven, using bioinformatics.
It means data-driven research means in your field you are doing some experiments, right? And making some new process and new solutions to address the problem of climate change related to some infections. So data-driven is the kind of approach we use data, and then from the data we can make some models, like in machine learning models, and to forecast some future for suggesting new approach or new techniques. So are you planning to include this kind of data-driven methodology or solution in your research?
You mean the data of the surveillance?
Yes.
Oh, I'm sorry. We are not take charge of the surveillance, so we don't know. Sorry.
Okay, all right.
Thank you very much. So now the last speaker. Thank you very much.